Hello students, now we will deal with other topic, that is packaging of DNA. Means DNA, how it is embedded or compressed or how it is arranged in many folds inside the chromosomes. To know this packaging of DNA, we need to know some basic points regarding length of DNA. So, length of DNA is usually defined with number of nucleotides or number of base pairs. For suppose human being DNA is the length of human double-stranded DNA if we want to calculate the length of a diploid cell, which is length of DNA, double-stranded helical DNA in a diploid human cell, then there is a small formula, that is we can apply number of base pairs into the distance between two nitrogen base pairs. Distance between two nitrogen base pairs. Number of base pairs in a diploid human cell, its number is 6.6 into 10 power 9 base pairs into what is distance between two nitrogen base pairs. If we draw the helical structure like this, here we are telling 10 number of base pairs are present in one complete turn of double-stranded helical DNA, that is 0.34 into 10 power minus 9 base pairs. If we calculate this one, we get approximately 2.2 meters. So in this way, by using such type of formula, we can calculate length of any type of living organism. We will see some other examples regarding the base pairs that are present in different types of organisms. In the same way, we can also write length of DNA in E. coli, that is calculated as 1.36 nanometers. A bacteriophage, here we can take example 5X174. It has 5,386 nucleotides. Another bacteriophage. A bacteriophage, these are all examples or some details given in your NCERT textbook. That is lambda phage has 48,502 base pairs. Next, how many number of base pairs are present in an E. coli? E. coli or Escherichia coli, it has 6.6 into 10 power 6 base pairs. Next, haploid content of DNA in human cell, or we will write of human DNA, that is 3.3 into 10 power 9 base pairs. These are the standard values regarding the number of base pairs and the number of nucleotides and also the length of DNA. So DNA is very compactly arranged inside the chromosome with support of some types of proteins called as histone proteins. Then when DNA is coiled around such types of proteins, then it is giving out different types of names like nucleosome, solenosome, solenoid, etc. You will see some of the terms like that. Now, we know that DNA is compactly coiled around different proteins called histone proteins. Histone proteins are permitted only to eukaryotes. But in case of prokaryotes, there are special types of proteins called as polyamines. So here, we know that one important point, DNA is acidic in nature. Previously, we have discussed this word, acidic in nature. Then how it is arranged around the proteins? And that acidic nature is due to many number of phosphate molecules. And its ends or it is coiled around the basic proteins, or it forms an attachment with basic proteins. It means that proteins generally are basic in nature. Now, in prokaryotes, the DNA is coiled using non-histone proteins called polyamines. But in case of eukaryotic cell, we have histone proteins. The compactly arranged DNA in prokaryotes, because of non-histone proteins, that is polyamines, it is also called as nucleoid or genopore. The compactly arranged DNA in prokaryotes, along with some gelatinous material around it. It is called nucleoid or genopore. Then what about eukaryotes? In eukaryotes, DNA is coiled around lysine and arginine rich histone proteins. It means that histone proteins are having more content of lysine amino acid and arginine amino acid comparing to other types of amino acids. In eukaryotes, the DNA is coiled around lysine and arginine rich histone proteins. Histone proteins are of 5 types. What are those? Those are termed as H2A, H2B, H3 and H4. Another one is H1. But these H2A are 2 in number, H2B are 2 in number, H3 histone protein are 2 in number, H4 histone protein is 2 in number. But H1 protein is only 1 in number. These H2A, H2B, H3 and H4 type of histone proteins altogether are called as Histone Optomer. These 4 pairs or 8 histone proteins together called as Histone Optomer. There is another name for Histone Optomer as new body, NU, not NEW. It is NU, new body, or it is also called as core of nucleosome. core of nucleosome. So it is confirmed that the negatively charged DNA is attracting the positively charged histone proteins. The positively charged ends of Histone Optomer is directed outside and so they attract the negatively charged DNA particles. Now we will see the structure of this histone optomer and arrangement of DNA around histone optomer. H2A, H2B, H3 and H4. So H2A, 2 number means pair, H2B and H3 and H4. So this is H3, this is H4, this is H2B, this is H2A. So totally 8 number of histone proteins. Now DNA is coiled around histone optomer like this. And again it continues with another coiling of another histone optomer. Then where is the existence of H1? H1 is outside the coiled complete structure. This is H1. This complete structure of histone optomer plus DNA is called as nucleosome. Nucleosome. Now, nucleosome is also called as bead in string. We also call nucleosome with another term, that is bead in string, form of chromatin. Form of chromatin. A nucleosome, typically in case of living organisms, it has, we'll write, typical nucleosome has 200 base pairs. Means a typical length of a nucleosome can be considered with 200 base pairs. But in human cell, nucleosome has 146 base pairs. The length of DNA in case of nucleosome of human cell is having 146 number of base pairs. This can be considered as the length. Out of this 146, or in a typical nucleosome, only 3/4 part of DNA is coiled around histone optomer. The DNA is only 1/3/4 part around histone optomer. Means if we consider the total 200 base pairs in a typical nucleosome as one complete unit, out of that only it means that 75% is only coiled around histone optomer. And the part that is between two nucleosomes, if we consider this one as one nucleosome and this as another nucleosome, means bead-like structures, the distance between, or the part of DNA between two nucleosomes, we can consider here this one as linker DNA. It is called linker DNA. Again, I am repeating, out of the total length of this 200 base pairs or 146 base pairs, only 1 3/4 part of DNA is coiled around histone optomer. DNA between two nucleosomes is called linker DNA. And the length of linker DNA can be 2 nanometers. We know that complete length of DNA in case of a human cell, it is 2.2 meters. But the linker DNA's length is very much less comparing to that of complete length of DNA. Size of nucleosome can be said in other terms, that is of 110 into 60 angstroms in length. This one can be said in terms of nanometers as or 11 into 6 nanometers. In case of angstroms, it is 110 into 60 angstroms in length. In terms of nanometers, it is 11 into 6 nanometers in length. The length of linker DNA varies from species to species. Why we are calling nucleosome as bead in string? Nucleosome appears as bead-like structures under electron microscope. Now, in terms of nanometers, what is length of nucleosome? Nucleosome is of 10 nanometers in length and the term nucleosome is given by Audet. It is given by Audet. Next. Nucleosome is found as thickenings of chromatin by Woodcock. Found as thickenings of chromatin by Woodcock. Here scientist names are very much important because sometimes while framing out the questions in previous MCQs, they are giving the correct scientist or the person contributor name, but along with that correct option, other 3 options are being included with some types of scientist names like this. So, in this chapter and even in Principles of Inheritance chapter, it is better to know about contributors or scientist name also. Now, when many nucleosomes are present side by side and when their length has reached 30 nanometers, the same beads or the nucleosomes are given with another term, that is solenoid. Here we have written the length of nucleosome is 10 nanometers. When the length of nucleosome, not nucleos, I am writing here about nucleosomes, not about one single nucleosome. Nucleosomes together has reached 30 nanometers in length, then called as solenoid form. then called as solenoid form. Now the length changes, that is 30 nanometers in length. We can show this one in diagrammatic representation. If you say this one has one nucleoid with histone optomer, like this, then if it is considered as 10 nanometers, one nucleosome, then if its length has been reached 30 nanometers, then we can represent the existence of nucleotides as nucleosome as like this. Means there are many number of nucleosomes, 1, 2, 3, 4, 5, 6, 7, many nucleosomes, but again they are intercoiled around itself. Then obviously the length changes. Then it is called as solenoid form. When this solenoid is highly packed and highly coiled around itself, then it forms another name called as super solenoid form. It means that the solenoid form is highly coiled around itself. The highly coiled solenoid form of chromatin or solenoid fibers, we write, forms into super solenoid form. Now again the length changes from 30 nanometers to 300 nanometers of length. Now this super solenoid form is further condensed or coiled. Condensed or coiled to form a new structure again in which the length and the term changes. A chromatid. Chromatid is having how many nanometers of length? It has reached from 300 nanometers to 700 nanometers of length. Now, further condensation is leading into formation of chromosomes. Here it is an MCQ. Further condensed or further condensation happens at metaphase of cell division, forming into new length structure, that is chromosome, forming into a chromosome. Now complete chromosome, now its length also will be changed, that is 1400 nanometers. Means the total DNA or the nucleosomes or the solenoids, their length will be changed, and also the terms that are given for those highly coiled structures now is of 1400 nanometers in length. In case of higher organisms, an additional set of proteins are present along with histone proteins. Those are called as non-histone chromosomal proteins. Just we'll write the simplified form, that is nucleosome, which is a composition of DNA plus histone optomer plus histone optomer, this one. Its length is 10 nanometers. It is condensed to form into solenoid, which is of 30 nanometers. Again, it is further condensed to super solenoid, whose length is changed into 300 nanometers. This is again condensed to form into chromatid, whose length is 700 nanometers. It is further condensed and formed into a chromosome. Now the length is 1400 nanometers. Here we did not include the linker DNA, but from here onwards, between two nucleosomes we are telling. So here there is no point of H1 histone proteins, but from here onwards, basing on the length, we used to include H1 histone proteins from solenoid part. In higher level organisms, there are some different types of proteins which are acidic in nature. There is presence of acidic non-histone proteins, non-histone chromosomal proteins. These are written with shortcut, that is NHC. So NHC or non-histone chromosomal proteins are of three types. What are those? One is structural proteins or scaffold proteins. These are made up of approximately 20 number of amino acids. So here we can write one example as topoisomerase, which we read in DNA replication, which happens in the S phase of interphase just before mitosis or meiosis. Commonly we used to say as preparatory phase. Next is functional non-histone chromosomal proteins. Here we can write examples as DNA polymerase or RNA polymerase. These are working in the form of enzymes. Other type of non-histone chromosomal proteins are regulatory proteins. The function of these regulatory proteins is they bind with DNA and they change complete the structure of concerned genetic material. Regulatory proteins, they bind to DNA and nucleosomes and change the structure. They are also responsible to control gene expression. They control means they are causing changes in genetic information also. They control gene expression. These are also called as high mobility group of proteins. Also called as high mobility group of proteins. Now, in case of chromosome, if you see the structure of X chromosome like this, the center part we know, we call it as centromere. Here will be the arrangement of highly coiled DNA. The nucleosomes, the solenoids, chromatin, the bead-like structures. So everything is highly coiled. So here only the length changes from 10 nanometers till the formation of 300 nanometers in different types of arrangement of this. uh, nucleosomes. Now, this part is loosely arranged chromatin, and it is considered as functional chromatin. It is involved, this part of chromatin is involved in transcription, translation. Is functional and has genetic information. So this part, it can be this or this or this or this. This is called as a euchromatin. It is responsible for transcription and translation process and transfer of genetic material also. Sorry, genetic information also. This red color labeled structure is also the chromatin, but it is highly thickened type of chromatin. This is called as heterochromatin. Heterochromatin is tightly packed and dense chromatin. We call this structure as bar body. Means this part. Because of this part only, we say X chromosome of a pair of females. In case of females, they have bar body. Bar body term is because of existence of this highly densely packed heterochromatin part. Because of this heterochromatin only, the term is given for one of the X chromosome of the pair as bar body. The stain used to find heterochromatin, or normally another sentence can be framed, the stain that is used to see the chromosomes also, Giemsa stain. Heterochromatin is Giemsa stain. Now, you will write another point. I think I have written here is functionally active, transcriptionally active. Euchromatin is transcriptionally active. When we add Giemsa stain, this part appears light in color. Light in color. We can also say the light colored strands. How much percentage does this euchromatin exist in the chromosome? It is of 30% of total DNA in a cell. You can also say it is of 30% of the genome. Genome means the genetic material inside one cell. Coming to heterochromatin. Heterochromatin constitutes of is of 70% of total DNA in a cell. Next, when it is stained with Giemsa stain, it is appeared in dark color. Appears in dark color. Next, this one is transcriptionally inactive. This is about the DNA, its length, presence of DNA in the form of nucleosomes, coiling around the histone proteins, presence of DNA in the form of solenoids, supersolenoids, chromatids and chromosome. Now here, I want to just mention one more point, that this part is called as arm. This part, it is called as arm. This is also called as another arm of the chromosome. So this is also another arm of the chromosome. This is also another arm of the chromosome. So in an X chromosome, there are how many arms? Means 1, 2, 3, 4. Then what is chromatid here? If we connect two vertically arranged arms, Then this part along with one centromere, this is called as chromatid. In the same way, this part, uppera, lowera and little bit part of centromere, this is also called as chromatid. Completely we are calling as chromosome. So 4 arms, 2 chromatids, 1 chromosome. So this is about the DNA till the formation of one complete chromosome structure.